Welcome to our webinar series 3.0 organized by Center of Health Science and Technology, JIS Institute of Advanced Studies and Research, Kolkata. I am Devavani Ganguly, today's host. Before I introduce today's speaker, let's watch our video together to know more about our institute.
A vision emerges in the mastermind of Padmasri Professor Ajay Kumar Ray to develop a unique postgraduate research institute with a difference, promoting an interdisciplinary approach, offering translational research-based education, and focusing on areas with high-quality job prospects and entrepreneurial opportunities. Fortunately, JIS Group educational initiatives joined hands in this dream project. And then, in mid-2019, under the aegis of JIS University, JIS Institute of Advanced Studies and Research Kolkata was born. With 3 centers, Center for Data Science, CDS, Center for Health Science and Technology, CHEST, and Center for Interdisciplinary Sciences, CIS. Now, let's dive deep into the CHEST. The demand of technological expertise in both academic and industry sectors of health science can only be ever increasing. On one hand, there's a continuous need for biotechnological developments, especially for direct medical applications. Simultaneously, the advent of newer high-throughput technologies asks for powerful bioinformatics solutions on how to extract, tackle, analyze, and visualize the biologically meaningful data for subsequent research and industrial applications. This scenario puts qualified bioinformatics professionals, especially the ones with expertise of analyzing medical biotechnology data, are in high demand for top salary jobs worldwide. Keeping this in mind, CHEST offers 2-year MSc in Medical Biotechnology and Bioinformatics, 2-year MTech in Bioinformatics, and regular as well as integrated Master's PhD program in related fields. But why would you select CHEST? Here are 3 reasons why CHEST is the best nest to hatch your career flight in health and medicine. Number 1. State-of-the-art molecular genomics, biotechnology, and bioinformatics laboratory with avant-garde equipment like PCR, real-time PCR, tape station, gel documentation system, CO2 incubator, fluorescence and UV spectrometer, biosafety cabinet, fume hood, and many more, along with ample laboratory workspace. And finally, last but not least, Illumina MiSeq, one of the highest ranked platforms for genome sequencing. JIS IASR boasts to be the only private educational institute and one of the very few among all public plus private institutes in Eastern India to have the in-house facility of this high throughput next generation sequencing and data analysis. Number 2. Internationally experienced faculty team with prestigious research grants, top-tier journal publications, and global collaborations. Multifaceted domain knowledge of CHEST faculty members aims a paradigm shift in infectious diseases research in India. The expertise in the CHEST faculty team spans from molecular microbiology and host-pathogen interaction, adaptation strategies of microbial pathogens, evolution of microbial virulence and antibiotic resistance, molecular modeling to explore functional insights of biomolecules, to as diverse fields as image processing and machine learning, as reflected via seminal publications in top-tier journals like Cell, Nature, Nature Light Science and Applications, Nature Protocols, Nature Communications, Journal of the American Chemical Society, PLOS Computational Biology, Proceedings of the National Academy of Sciences USA, to name a few. This has been possible through worldwide collaborations with academia and industry and prestigious research grant funding from Government of India and Wellcome Trust in United Kingdom, an unparalleled achievement of an Indian institute within the first year of its inception. Number 3. Need of the hour biotechnology and bioinformatics programs in health and medicine. Along with a solid foundation of different aspects of medical biotechnology, computational biology, and bioinformatics applications, our MSc and MTech programs include some of the key subjects of utmost industrial needs. For example, next-generation sequencing data analysis, microbial virulence and antibiotic resistance, proteomics and metabolomics, computer-aided drug design, pharmacoinformatics, systems biology and biological networks, big data in bioinformatics, and so on. And expertise in any of these fields can readily offer lucrative jobs in pharmaceutical and biotechnology industries, in hospitals and clinics, in bioinformatic data analysis companies in India and abroad, besides the standing opportunities of research and academic positions. Apart from regular subject-oriented topics, our programs consist of topics like research methodology, career development and entrepreneurship, and intellectual property rights. This edition not only enables the students to cope with the situations in industrial or academic environments during their summer internships or final semester projects, or in their job positions after the course completion, but also aims to motivate the students to develop an entrepreneurial mindset. So, why delay? Join CHEST, the best nest to hatch your career flight in health and medicine, and target the top together.
Thank you for watching. Our today's speaker is Dr. Arunima Sengupta. The title of her talk is Cardiomyocyte Survival and Death Pathways, a fine balance in development and in disease. Dr. Sengupta got her PhD in molecular physiology in 2007 from Miami University, Ohio, USA. Then pursued her postdoctoral research training from Department of Molecular Cardiovascular Biology, Cincinnati Children's Hospital Medical Center. This is also from Ohio. After returning back to India in 2014, she spent a year as faculty in Akash Institute, South Calcutta. She received her prestigious BioCare Women Scientist grant and worked for an year in Department of Genetics, University of Calcutta. In June 2016, she has joined Department of Life Science and Biotechnology in Jadavpur University as UGC Assistant Professor. The main objective of her research is to study how the several extracellular pathological, hypertrophic and fibriotic, my mistake, simulations like hyperglycemia, pollutants is perceived and converted into intercellular signals, and how these signals eventually leads to cardiac problems. So let's introduce Dr. Sengupta, and we are from on behalf of JIS IASR. I am extremely thankful to her for accepting our proposal or our invitation for this webinar. And now, Dr. Sengupta, the stage is yours. And for the participants, for the candidates who are not the candidate, my mistake again, the attendees here, please, you can write your questions in the question answer Q&A section or in the chat sections. After the talk, Dr. Sengupta will address all the questions. Thank you very much for attending, and please, Dr. Sengupta, you may start. I'm stopping this screen share, and also you can have all the detail in this our Facebook page, https://www.facebook.com/JISR. Okay, so now let's start the talk.
Okay, a very good afternoon to all of you. This is Arunima Sengupta. I'm from Jadavpur University, and as Dr. Devabani Ganguly mentioned. Now, first of all, I would like to thank the organizer for giving me this opportunity to share my research work and my thoughts, what I'm thinking, how I'm approaching all the problems related to my research area. And special thanks to Dr. Devavani Ganguly for contacting me and inviting me to for the talk. So let's start. Okay, so the title of my talk is Cardiomyocyte Survival and Death Pathways, a Fine Balance in Development and Disease. Now, what I have decided for today's talk, that I will not show you many data, what I have with me, but I will give you an overview or a review kind of thing, that what the current understanding of a specific aspect of cardiovascular or heart development and disease. Now, as you can see, that the mature mammalian heart is a very complex structure. And if we start from our developmental point of time, basically the heart is the first pulsatile organ of an embryo within the utero. That means this is the first organ that starts functioning when the baby is within the mother's womb. So you can imagine that once it started, basically that pulsatile thing, it needs to do its job throughout individual lifespan till his last breath. Now, this mature heart, this adult mature heart, is made up of many different kind of cells, okay? Starting from myocardial cells, endothelial cells, smooth muscle cells, fibroblasts, specialized conducting cells. These are the few that I mentioned. There are many more things. So what I wanted to point out over here is that it is a very complex structure, and there are many cells that are present within the heart. It's not only the cardiac muscle cell or cardiomyocytes that we think, but there are many other cell types that are present, and all these cell types are performing their function individually, as well as they are coordinating with each other to make the functional heart. Now, this is another figure of the same thing that I have mentioned. Now, this was like a schematic diagram, but this is like a section of a heart. If we, the mammalian heart, if we do a section and then we do simple H&E staining, you will see that this is the fourth chamber, these two are the atria, and this is the ventricle, this is another ventricle, and this is the interventricular septum. These are basically the lumen, and these are the valves and etc. So what I wanted to point out again over here, you can see that there are many different cell types like cardiomyocytes, conduction specific cell types like node cell, Purkinje fiber cells, etc. Fibroblast, another very important cell types that is present within the heart. Then epithelial cells, vascular smooth muscle cell, endothelial cells, and cardiac melanocytes. Now, if we go back to the development again, basically this heart development start with the formation of this cardiac crescent cells. It's like a crescent shaped structure, and then following the morphogenetic movement and the differentiation, basically these cells transform into a linear heart tube. Then it forms a looped heart, and finally we are getting this four-chambered mature heart. So my point over here again is that the simple crescent-like structure is basically transforming with different movements, differentiation, and other processes to form this mature heart. So all the events that are taking place during the development is very crucial for the functioning of the adult heart. Now, in today's talk, basically what I will do, I will mainly talk about the cardiomyocytes and little bit of the fibroblast cells, because it's not possible to talk about all the different cell types. So the main cells that is present within the heart is the cardiomyocytes, and there is another population, it's the cardiac fibroblast. And I have chosen two different aspects. One one during the development, what is happening, and another during the disease, what is the main thing that is happening. Now, first I will start my talk with the developmental time point, where I will mainly focus one aspect of cardiomyocyte, and that is the growth. One thing you can understand from the figure, that a linear, a crescent shaped structure is forming a fish, like a size of a fist, the mature heart, right? So there is a tremendous growth of the heart. Now, how this happens basically? Now, there are two phenomena. One is the hyperplasia or the proliferation. That is the simple division, mitotic division of the cardiomyocytes, and giving rise, the growth is happening. There is another phenomenon which is very unique to this cardiomyocytes, and there we can see some other cell types also, that there is an hypertrophic growth. So what I wanted to point out, that these are suppose the normal cell. Now, hyperplasia or proliferation, meaning if you can see the bottom panel, basically one cell is giving rise to two to four, 4 to 8, like that. So that is basically we are increasing in the number, but the size and the shape of the cells remain same. Whereas in case of hypertrophic growth of a cell, meaning when there is an increase in the cell size instead of division. So there is no division occurring, but the size of the cell is getting bigger, and that is known as hypertrophy. Now, why I mentioned over here? Because now during this developmental time point, there is tremendous rapid rate of cardiac proliferation or hyperplastic growth. That is the cell division. But soon after birth, during the neonatal time point, and there is a switch from this hyperplastic growth towards the hypertrophic growth. Okay, so there is basically gradual decrease in the rate of cardiac proliferation. That is, the rate of proliferation get decreased, but the cardiomyocytes is now getting bigger with the help of hypertrophic growth. Okay, so that is, the cell number almost remains same, but the size of the cells getting increase. So the burning question is that this switch, what are the different molecules that basically involved in this switch from the hyperplastic growth to the hypertrophic growth? That was the main question. When I started my career as a postdoc, basically I had this question in my mind, like during developmental time point, what are the different cues that basically helps the cardiac myocytes to switch from this hyperplastic growth to hypertrophic growth? Now, these are basically the review paper. There are plenty of review paper on cardiac proliferation and growth. As you can see, if you consider this as the developmental time point, E meaning embryonic, so embryonic 10, P0 is just at birth, P5 is postnatal day 5, so after 5 days of birth, 14 days, and then adult. This rate thing is basically the rate of proliferation. You can see over here. Okay, now you can see that during the embryonic time point, there is a tremendous proliferation of the cardiomyocyte, and there is a sharp decrease postnatal. Then we can see a little increase, but after that there is a gradual decrease, and very interestingly, the adult cardiomyocyte has very little to no power of proliferation. That means those cells are almost terminally differentiated. Although I cannot say that 100% cells are terminally differentiated, there are stem cell population that are there, but that is a different topic. But majority of the cardiomyocytes are basically fail to re-enter into the cell cycle. They basically goes into the senescence, kind of stop their division. Okay, and mainly the growth occurs through hypertrophic growth. So here you can see, so my question is basically, what are the signals that determines this switch? What's the importance of that? Now, the same thing basically, now fetal cardiomyocyte, they are highly proliferative. Few signaling molecules are already published out there, neuregulin, ARB2, TBX20, BMP, IGF-PI3 kinase, etc., etc. But the switch, like from this, all these are very important for the cardiac proliferation. Now, what's the cue which helps this proliferating cardiomyocyte to form a mature cardiomyocytes with only hypertrophic growth? Now, why this is so important? Why should we study this? Now, The inability of the adult cardiomyocytes to re-enter into the cell cycle is one of the limitation of poor cardiac regenerative potential. Now, as you can see that basically, I told you that the adult cardiomyocytes are they don't divide. So what happened? Like we are familiar the terms with the terms like heart attack, right? Or myocardial infarction, heart failure. These are the terms that we get to hear from different doctors or some. Now the thing is, what happened during this heart attack? Heart attack is basically is the death of cardiomyocytes. Now you can imagine that in adult heart, the main function of the heart is to pump the blood, right? To pump the blood so that the blood can circulate to the different parts of our body, and blood contains all the nutrients, all the oxygen and everything, all the goodies that we need for our cells to grow, right? So that is main thing. Now, in case of heart attack, basically there is sudden loss of cardiomyocyte, death of cardiomyocyte. As a result, I told you that adult cardiomyocytes fail to divide. They have very little to poor regenerative potential or the divisional capacity. So as a result, if you lose the cardiomyocytes, there is no way you can replenish that cardiac cells, because they don't proliferate. So in that case, it's a problem. Now that individual has to survive with whatever the cardiomyocyte he's left after the heart attack. Now, if it is a massive, then there will be some fatality also along with it. Now, therefore, it is very important to know what are the signaling pathways that basically helps the cardiomyocyte to proliferate. And if we know that, and if we know the signaling pathway that can switch this hyperplastic to hypertrophic, maybe we can exploit this pathway later in the adult injury to make the cardiomyocyte again come back into the cell cycle and start divide. And that in that way, what will happen, we can regenerate the cardiac cells so that they can perform and they can efficiently circulate all the blood. Now, there is a difference between heart failure and heart attack, right? Heart attack, I just mentioned you that it is the sudden death of the cardiac myocyte. Now, heart failure is basically like the what we use generally when particular job is not done. So the main function of the heart is to pump the blood. So heart failure is basically, it is a compromised heart which is not able to circulate the blood efficiently like a normal individual. Therefore, what we can say that this heart failure basically it is basically inability of the heart to circulate the blood efficiently. Now, why I am telling all these things? Because the whole point of my my first part of my talk is basically to understand the signaling, so that we can use those and we can exploit in effort to reactivate the proliferation of cardiomyocyte in treating cardiac disease. Now, this is an immunofluorescence study basically to show you, to give you an perspective what we do in the lab. Basically, these are the sectioning of the heart, the mouse heart. Okay. Now, this is E9.5, meaning embryonic 9.5. After conception, it is a 9.5 day embryo. Basically, the pups we harvest, we do the sectioning of the heart, and then we are staining with specific antibody. This is 14.5. That means after embryonic day 14.5, and this is a 4-month-old adult. Okay. Now, in the thing that I wanted to point here is the pink staining. Now, you can see those pink dots. These are nothing but the indication of that the cells are dividing. So it's a proliferation marker. Okay. Simply, if you just look the picture, one thing you will notice that the pink staining is gradually decreasing. The highest is over here, then it's decreasing, and finally in the adult we can see very few, and these are not even cardiomyocyte, because the green staining indicates the cardiac myocyte, and these are the other kind of cells that are proliferating, but the adult cardiomyocytes are not proliferating. So again, the bottom my message is that the adult cardiomyocytes does not proliferate. Now, there are like a group from Houston, the SADC, they have done a beautiful experiment where they have used adult zebrafish, adult newt, and fetal mouse, fetal or neonatal mouse. And what they have done, they have basically injured the heart. Okay, after injury, what they have noticed that in these three cases, the first three, the adult zebrafish, adult newt, and the mouse. Okay, in these three cases, the cardiomyocyte can undergo proliferation and they can regenerate. But in case of adult mouse, after injury, they are hypertrophic rather than hyperplastic, and they fail to regenerate. So one thing we are sure, and in human is basically, in human is same as the mouse, that there is no regenerative potential in the adult cardiac myocyte. So the burning question, why the zebrafish, neonatal mice, and then newt, they can do the regeneration, but why not? And these are the $1,000,000 question that many scientists, group of scientists, they are working. They are trying to find out how we can make this stubborn adult cardiomyocytes in mice, in mammals rather, and make them to re-enter into the cell cycle so that they can grow. Now, when I started my work, basically, I got interested in this aspect, and one molecule that interested me was the FOXO transcription factor. Now, when I started, it has many. This is a transcription factor, so you can imagine that it would bind to a particular genomic sequence, and then it can induce the expression of several target genes. It has many functions, but very limited information was there regarding its role in the cardiac cellular proliferation or hypertrophy. Okay, one thing is that when there is a growth factor limitation or nutrient depletion or hypoxia, there is increase in the FOXO expression. So that was known. So the first part, basically, I will talk about the development, that how FOXO regulation of neonatal cardiomyocytes, whether there is any role in of FOXO or not. Now, in fetal myocardium, what we know that FOXO is very inactive, very faint expression we can see. But in neonatal cardiomyocytes, neonatal means just after birth or adult, there is moderate level of FOXO expression. So that gives me an impression, since during development there is very little expression, but suddenly there is an increase in the expression of FOXO in the postnatal, maybe this FOXO can have some role in the regulation of this hypertrophic to hyperplastic to hypertrophic switch. And from other cell type, it was known that, and when FOXO is inactive, there is high level of proliferation. So that fits with my hypothesis. Maybe since FOXO is inactive, the myocardium is highly proliferative, and then when FOXO is more in the adult one, maybe that actually inhibits the proliferation, maybe help in hypertrophic growth. Now, before I want to introduce two more molecules to give you, I will not show you many data, what I have done, but just to give you how we approach a particular problem, and then what kind of experiment we do to answer those questions to give us a conclusion. One kinase got my attention is the AMPK activated protein kinase. And why? Because I have noticed that AMPK can directly activate FOXO in many different cell types, where nothing was known in cardiac myocytes. And importantly, AMPK also inhibit cardiomyocyte proliferation. So I thought whether AMPK can also regulate FOXO in the cardiac myocytes to withdraw the myocytes from the cell cycle or not. And another molecule also I got interested is the FOXM1, where basically I know that this FOXO and FOXM1 actually inversely regulated. So when there is increased expression of FOXO, there is decreased expression of FOXM1. When there is increased expression of FOXM1, there is decreased expression of FOXO. So like altogether, basically, I was working with these three molecules to test whether there is any role of these molecules during this neonatal to adult transition or not. And the thing is that both AMPK, FOXO, I told you they are very low levelly expressed during the developmental time point, but there is sudden increase in the adult. Whereas in case of FOXM1, the expression is completely reversed. FOXM1 is highly expressed during the developmental time point of the cardiomyocyte, but gradually decreases. Now, based on all this, I hypothesize that probably in the neonatal cardiomyocyte, since there is growth factor limitation, there is activation of AMPK which activates FOXO, and maybe that deactivate FOXM1. As a result, since FOXO is an inhibitor of cardiomyocyte proliferation, which I am not showing here because its direct transcriptional target is P21 and P27, which are the inhibitor of cardiac cell proliferation. So maybe that is the reason that this AMPK and FOXO, when you have too much that in the cell, that inhibits the proliferation. And basically, I hypothesize maybe FOXO is directly regulate FOXM1. So I told you that in other cell type, it has been shown that FOXO, when you have more FOXO, then FOXM1 goes down. When you have less FOXO, FOXM1 goes up. Maybe that is also happening in the cardiomyocytes with the growth factor. The IGF-1 is one of the growth factor to promote the withdrawal. So my overall hypothesis, AMPK mediated activation of FOXO transcription factor inhibits FOXM1, and the balance between FOXO and FOXM1 controls IGF-1 levels to promote cell cycle withdrawal in neonatal cardiomyocytes. Now, fetal cardiomyocyte, there is plenty of work in the fetal cardiomyocyte, and I told you insulin, neuregulin, these are the growth factor, AKT, PI3 kinase, these are kinase which can regulate FOXO to inhibit. I mean, when these are basically active, FOXO remains inactive, and you have normal cardiomyocyte proliferation. So the question that I asked, maybe in the postnatal cardiomyocytes, there is low level of growth factor, maybe that activates AMPK, which can activate the FOXO, and that can inhibit the proliferation or promote, I mean, inhibit the proliferation and promote the cell cycle withdrawal. Now, these are just a few data just to show you that these are the neonatal cardiomyocytes. These are the individual cells. The red staining shows you the cardiomyocyte staining, alpha-actinin. Okay, Ki67 again is the proliferating marker. This is a Marge even, and the TOPRO is the DNA dye, basically, and the nuclear staining. So you can see that in the control, you have very moderate level of proliferation. These are the neonatal cardiomyocyte. But when I inhibit AMPK, so compound C is an inhibitor, AICR is the activator. So when I inhibit, you can see there is almost all cells are proliferating. So the bottom line of this slide is basically that the inhibition of AMPK results in increased activity of proliferation of cardiomyocyte, whereas when you activate, you have decreased. Now, this results also correlates with the localization of FOXO. Again, this is another immunostaining just to show you the localization. This one is FOXO1, this one for FOXO3. So you can say that when compound C, I told you what compound C is the inhibitor of AMPK. And when it's inhibit, basically, I told you that there is increased in proliferation. Okay. Now, if you see that when compound C, we are adding compound C, the FOXO1, very less amount of FOXO1 is in the nucleus. It's mostly cytoplasmic. But when we activate, it's all are in the nucleus. See, everything, all the FOXO, they are in the nucleus. And same is true for here. You can see that these are the nuclear, but here almost everything is cytoplasmic. So the thing I wanted to mention here, that the localization of AMPK and FOXO, they correlates with each other. When there is more AMPK, there is FOXO in the nucleus. When there is less AMPK, the FOXO is also outside the nucleus and in the cytoplasm. And if transcription factor is outside the nucleus, increase or decrease the expression of the target gene. So now is the same thing I have shown you with the AMPK. Now I'm showing with the FOXO. These are the different FOXO adenovirus. Okay, the same thing. So when we inhibit FOXO, we see more proliferation. When we activate FOXO, less. So all these three figures, I will not show you any more data regarding this, but I just wanted to point out that what I have shown, that AMPK and FOXO is the inhibitor of the cardiomyocyte proliferation. Okay, because when I am activating, I am seeing decrease, but when I'm inhibiting, I can see more proliferation. The same thing, those were in vitro culture system, in cell culture, basically, neonatal cardiomyocyte, and these are in the mouse heart. Okay, these are the transgenic mouse heart, where basically I have deleted FOXO1 and FOXO3 specifically in cardiomyocytes, and I can see that there is increase in the proliferation. And this is the last data slide I wanted to, and then I did lots of other experiment to find out the exact hierarchy of this AMPK, FOXO, FOXM1, all those things, how they're, who is upstream, who is downstream, and who is activating what, those kind of thing. And ultimately what I have shown in this figure, that I found a common target, a novel target of FOXO and FOXM1, and that is the insulin-like growth factor 1. And what I have shown here, that I will not bore you with the data, but what I wanted to mention over here, that both this transcription factor, this FOXO and FOXM1, they can bind in the promoter site of IGF-1, but one is activating IGF-1 and another is inactivating IGF-1. FOXO will inactivate because this is the growth factor and that will promote the cardiac proliferation, whereas FOXM1 is a positive regulator of proliferation, so it activates IGF-1. Okay, and basically, to put all the things in a schematic way, what I have shown, that during fetal cardiomyocytes, since there is ample of growth factor, okay, there is ample of growth factor, there's plenty of insulin and everything. As a result, what happened, there is low level of AMPK, and I told you, if there is low level of AMPK, FOXO is also low, but this high growth factor promotes FOXM1, and when there is ample of FOXM1, it comes into the nucleus, it promotes IGF-1, and there is high level of proliferation. But in case of neonatal cardiomyocytes, when we have low level of growth factors, maybe this is the key, that then the AMPK level got upregulated. As a result, FOXO is also upregulated, and thereby the FOXM1, since it's a inactive form because low growth factors, so FOXM1 is low. Now what happened? The FOXO can bind to the IGF promoter. As a result, I told you that it inhibits IGF-1, so no IGF-1, and therefore the cells are not proliferating and they are withdrawing from the cell cycle. So this is a very small contribution from my part, like maybe this signaling pathway is one of the signaling pathway. Again, I'm not telling this is the only pathway. Okay, there are many other pathways, because in a cell we always take one particular molecule and we try to investigate, because it's difficult for us to do a global thing. But actually in the cell there are so many factors that are acting together. So again, I am telling you, this is probably one of the pathway that is actually resulting in inhibition of the cardiomyocyte proliferation and results in the withdrawal from the cell cycle and push the cell into the hypertrophic mode. Now I am moving to the second part of my talk, and that is the disease part. Okay, I have mentioned little bit about cardiac heart attack, heart failure, myocardial infarction. Now this is this cardiovascular disease is one of the leading cause of morbidity and mortality in not only in India and the worldwide actually. Now in estimated it is like 2.1 to 10.9, like 2.1 deaths and 10.9 hospitalization millions, okay, respectively by 2021. Now I told you one of the limitation is basically this death of cardiomyocytes. Now if there is a death, there is very little chance or possibility that those cardiomyocytes will again regrow. Okay, that was the inability of the myocytes to proliferate. Now what are the other way? The first thing is the cardiomyocyte loss. That's no question about it. Okay, second is the cardiomyocyte hypertrophy. I told you. Now hypertrophy is a normal physiological phenomenon, right? I'm telling you from the beginning. But in disease condition, it's called pathological hypertrophy, okay, and that is very bad for a cardiomyocyte. Normal physiological hypertrophy is fine, but pathological hypertrophy is bad, which ultimately results in fibrosis. Fibrosis is what? Basically, when there is a loss of cardiomyocyte, if there is a loss, you can see that there will be thinning of the cardiac wall, right? And if there is too much of loss, then that can rupture, and you can imagine what will be the end result. So So what happened? Our fibroblasts, the cardiac fibroblasts, try to take that space so that the rigidity, the structure of the heart remains intact at least. And as a result, the fibroblasts, they are highly proliferative, okay, in contrast to cardiomyocyte. So fibroblasts proliferate and basically replenish those region and form a scar, and that is known as fibrosis. See, what we have done, we have basically, whatever the loss of cardiomyocyte happened, we have replenish that with the fibroblast, okay, to maintain the structure of the heart. But will that serve the purpose? No, right? Because fibroblast, the function of fibroblast is not same as cardiomyocyte. Cardiomyocyte is the pumping cell that helps to pump the blood, whereas fibroblasts, they form the collagen, they form the extracellular matrix, okay, to give the rigidity, to shape, and there are some signaling function also. So basically, I am making the heart shape okay, but the function of the heart is compromised, okay. So as a result, what happened, we can see pathological hypertrophy, fibrosis, as a result cell death or apoptosis, and ultimately which can leads to heart failure, okay. Now this slide is basically again I am showing you the postnatal growth. Basically it's a normal physiological phenomenon that this heart, after hypertrophy growth, we are getting the normal size heart, and then it again ceases basically. Now during this time, if there are some insult can be outside the body, inside the body, diabetes, hormonal dysregulation, stress, toxicity, chronic hypertension, aortic stenosis. There are so many kinds of stimulus that can basically affect the growth of this cardiomyocytes. Now, what is the physiological hypertrophy? I told you, like during chronic exercise and during pregnancy. Like normally during pregnancy and exercise, what we need a lot of blood in our circulation, right? And that's why this is a normal physiological phenomenon that there is an intercell size of the heart, I mean cardiomyocytes. As a result, more bloods are pumped throughout the body, and we are getting more oxygen, known as the physiological hypertrophy. There is no fibrosis, no apoptosis, nothing. And when the baby is born, the pregnancy is gone basically, or when you are not doing the exercise, again the cardiac myocyte comes back to its normal size. But during pathological hypertrophy, okay, what happened? The myocyte getting bigger and bigger, and basically it crosses its threshold actually, and results in cardiac dysfunction, fibrosis, dysregulation of the calcium balance, apoptosis, necrosis, contractile dysfunction. There are so many things which can lead to heart failure, that is the myocardial cell death and everything, okay. So the hypertrophy is good in a way when it is physiological. When there is no, here you can see the sectioning, these are pathological. You can see that increase in the heart. If you compare this heart versus this heart, this is the pathological, and this is the physiological, sedentary heart and exercise heart. But this will again come back when the person is not doing the exercise. But the difference in the histology, if you see that in pathological, you can see a lot of blue staining over here, right? And these are the fibrotic patches. Basically, there is a death of cardiomyocytes, and that's get replenished with the fibrosis. But in the physiological exercise, see, there is no fibrosis and the heart looks fine. So my point is, and this is basically, it's a vicious cycle, okay. Once you have this heart failure, it goes on and on and on. Basically, there is less like nutrient depletion, ischemia, calcium overload, death, and reduced pumping action, and this will go on and on and on basically. So the question is, the burning questions that I have in my mind is that how these risk factors are perceived and converted into intracellular signals, and how the signals change the transcriptional program that eventually leads to cardiac hypertrophy, fibrosis, and apoptosis. So in my lab, basically, I'm doing two kind of stress. One is the environmental risk factor like arsenic toxicity, and the other one is the extracellular or internal risk factor like hyperglycemia, like when we have a high blood glucose level. And we are looking at a different downstream effector like hypertrophy fibrosis. We are seeing calcification, ear stress induced apoptosis. Now in today's talk, basically I will just mention these two, the hypertrophy fibrosis and ear stress induced apoptosis. Now very recently we published, my graduate student published a paper where she has shown that one of the signaling pathway that is basically upregulated in this arsenic induced cardiac hypertrophy is AMPK, FOXO and NFAT-C3. This is a novel thing that we discovered, that NFAT-C3 can actually, under the regulation of AMPK and FOXO, to induce cardiac hypertrophy. And she has also shown that the oleic acid, pre-treatment of oleic acid can actually protect this arsenic induced cardiac hypertrophy. So what I have shown here, again I will not show you many data, but just to give you an idea, that you can see in case of arsenic toxicity, this is, if it is a normal heart, you can see there is an increase in the heart rate and heart rate. Okay, these are the individual cardiomyocytes. See, you can see these are the control and these are the arsenic. You can see bigger cell compared to this one, and obviously this is the fibrosis. You can see in the arsenic there is blue patches compared to the control. And this figure is just to show you that oleic acid can actually reverse the phenotype. We do not have any fibrosis, we do not have any hypertrophy. You can see they are almost same, but this is the arsenic treated. So basically we can reverse the phenotype. And then the whole paper talks about the mechanism by which this oleic acid is actually reversing the arsenic induced cardiac hypertrophy and fibrosis. So one of the molecule that got highly upregulated is NFAT-C3. You can see this is the arsenic treated cardiac section of a mouse, and you can see NFAT-C3 expression. Again with the oleic acid pretreatment, we can see there is low level of NFAT-C3. And finally those were all in vivo in mouse, and this is again in the culture cell. Basically the arsenic results in increase, these are the phalloidin staining to show you the increased cell size. This is the basically again the NFAT-C3 expression. You can see this dark dot, these are all the NFAT-C3 positive, whereas in control only oleic acid plus arsenic can completely reverse. So the thing that I have showed you, that arsenic causes cardiac hypertrophy fibrosis, oleic acid can reverse it, and there is increased expression of NFAT-C3 probably if we can. So there are many experiments we did to find out the upstream effector, but I will just show you this data to nail down that the NFAT-C3 is the main culprit basically, where we have used either the NFAT-C3 protein, this is the recombinant protein data, and this is the INCA-C where we have inhibited the NFAT-C3. Okay, so again if you can concentrate in this figure, the arsenic treatment, you can see bigger cell compared to this one, the small one. Okay, now if we add INCA-6 to inhibit the NFAT-C3 and we add arsenic, we can see we prevent this hypertrophy. There is no such big cells. It's almost comparable with the control. And similarly, if we do NFAT-C3 recombinant protein along with oleic acid and arsenic, you can see that the cells are very big compared to control, in the sense over here almost arsenic treated cells and the size of the arsenic treated cells as well as this one. So the overall bottom line of this figure is basically to show you that NFAT-C3, if we induce the NFAT-C3 expression even in presence of oleic acid, we still see cardiac hypertrophy. And if we inhibit NFAT-C3, then we can inhibit the cardiac hypertrophy. And we have done some FOXO siRNA experiment to show, again to give an idea like who is controlling what. And ultimately from all these data, we have come up with this model where we are confident that arsenic toxicity is actually decreasing the AMPK and FOXO and thereby increasing the NFAT-C3 expression, which is results in cardiac hypertrophy. So one of the mechanism that arsenic toxicity results in cardiac hypertrophy through NFAT-C3 by inhibiting these two, whereas oleic acid is somehow activating AMPK and FOXO and thereby inhibiting NFAT-C3 and basically results in anti-hypertrophic effect. Okay. And this last few slides, the last slide basically, if I come back over here, so I mentioned this part. One of the thing that I chose is the arsenic toxicity, and this last one is this hyperglycemic toxicity. Okay. Now there are many paper that basically talks about how hyperglycemia can have adverse effect on cardiomyocytes. So the thing that I focused in my lab recently, I have started this, is basically to look on the ER stress response. Now, those who know that endoplasmic reticulum basically can detect whether the cell is under stress or not, then it can actually upregulate ER stress response called UPR, and then basically it has two arm. Okay, if there is mild ER stress or prolonged ER stress. If there is mild ER stress, then cell can undergo the cell survival pathway. But if the stress is too much and it's prolonged, then it can go to the apoptotic pathway. So my question, and this is a textbook from any cell biology textbook, we know there are three arms of the ER stress. One is the PARC dependent pathway, one is the ATF6 CHOP dependent pathway, and another one is IRE dependent pathway. Okay, I'm not going to talk about this path. I just wanted to mention you that there are many molecules, ATF6, ATF4, XBP1, CHOP, ERAD. So these are all the molecules that are involved in this pathway, and we generally look all the expression of these protein as a determinant of whether the cell is under ER stress or not. Okay, so we have just started to find out that is there any balance between the survival and death pathway, that if we tip the balance towards the survival pathway, is basically the cell can go into the survival pathway or not. Okay, so in order to do that, one thing we have noticed that this is again in the cell culture, where we have treated the cells with high glucose cell versus the low glucose one, and we have seen that under high glucose, you can see that all the ER stress markers are upregulated along with our molecule of interest, TBX20 and BMP2. Now we all know from the literature that TBX20, BMP2, they are very good molecule, and they are help in cell proliferation, they help in healthy survivability of the cell, those kind of things. So why in the hyperglycemic condition that there is increase in this two molecules? And this is the TBX20 expression. Again, you can see that this is in the mouse heart section. You can see there's a highly upregulated TBX20 expression in the cardiomyocytes compared to the control. So this is in the diabetic mouse. Okay, this is again in the cell. Now here, instead of glycemic hyperglycemia, we have used tunicamycin, which is basically ER stress inducer. Okay, now here I wanted to point out that I have used, or rather my students, she has used 10, 20 and 50 microgram of tunicamycin. And the thing that we have noticed, and it's a very preliminary data, that we can see there is increase in the TBX20 in the 10 microgram, but gradually there is again a decrease in the TBX20 expression. And if you can see that the proliferation marker Ki67, remember if I showed you before also, that when there is a 10 microgram of tunicamycin, you can see lots of cells are proliferating, but with 20 and 50, hardly, they're probably almost same as the control. So this gives an idea that if the stress is too much, then the cells are probably committed to the death pathway. Cell has no option. It cannot come back to the proliferating pathway. So that was our whole idea, how we can tweak this, how we can tell the cell to stop the stress response pathway and basically tip the balance towards the survival pathway. So I will basically end up here by giving you a hypothetical model that is in our mind currently. Basically this ER stress, if it's mild or prolonged, in case of mild, maybe we have the upregulation of the good molecule like TBX20 BMP, which is results in the cell proliferation and homeostasis. But if it is prolonged, and if we do not have this activation of this TBX20, maybe the cells is under the stress and going apoptosis and cell death. So these are very preliminary, and we will study further when things got normal. I guess we are really going through a trying situation over here. So I hope with this I end my talk over here, and maybe I have given you an overall idea that how mainly I have talked about the cardiomyocytes and fibroblasts, how these cells are working, and there are so many aspects in the heart basically which coordinately they are doing their job to make the heart function properly. I must acknowledge my students who are doing the job. Joita, who has published the arsenic thing, Shreya is doing the ER stress, and Arunima is doing the other thing that I have not mentioned. And my lab is funded with the UGC DBT grant as well as the RUSA from JU. And my collaborator, who has always helped me in many experiments, doing experiments in his lab, is Dr. Shantanu Chakraborty from Presidency University. So I will end up my talk over here, and I will be more than happy to take any questions if you have. Thank you. Thanks once again to the organizer of the JIS University for giving me this opportunity.
Thank you very much, Arunima, for this wonderful talk. There are many. Thank you, ma'am. It's a wonderful presentation. Got so many things known. So there are many things. But
I have to stop share, right?
yeah.
Thank you, Devani. Yeah. Tell me now. Yes.
So
thank you.
There are a few questions.
Okay.
The first one is from Procheta Pal. She asked, is the shift from hyperlysis to hypertrophy a reason why cancer does not occur in heart? Is there
very good question.
Yeah. And there is another question. Is there any chance of cancer in heart in embryonic stage when cardiomyocytes undergo proliferation?
A wonderful question. Okay, we see this we never heard cancer in heart, right? Have we? At least I have never. So it's a very unusual phenomena. I mean, that's why I guess, I mean, I always had this question when I used to do my postdoctoral study, that why heart, why there is no cardiac cancer? It's not that I want, but I had this question, like, and I think it is because of the nature of the cardiomyocytes, that it is so stubborn. Once it reaches its neonatal time point, there is no way you can push the cell again back into the cell cycle. I mean, you can, but that is like 1 in 10,000 arbitrarily. I don't know the number exactly, remember, but it is so less that so that's why all the scientists are actually looking for all the inhibitors. Maybe there are so many inhibitors which are still unidentified, and they are there in the adult myocytes which are not in the embryonic, and these inhibitors are actually preventing the cardiomyocytes to re-enter into the cell cycle. That is one possibility. Or maybe it is because the positive regulators are completely shut down. It's basically the two sides of the coin, right? Either the inhibitors are so, I mean, it's expressed in so abundant that there is no way that the cell cycle can progress, or the positive regulator like the kinase and others are inhibited in such a way that the cells fail to proliferate. And embryonic cancer, I have never heard, but there is some congenital defect, irregular or dysregulation of this signaling pathway can have adverse effect. But I don't think there is any term associated with heart and cancer. So I will not say it's embryonic cancer, but it is dysregulation of this proliferation pathway can have adverse effect on the functional outcome of the heart, the cardiomyocytes, pumping ability.
Okay, so yeah, the next is from Shubhashish Indra. Shubhashish wants to know, ma'am, if we provide sufficient amount of nutrients and the growth factors to neonatal heart, will they undergo division?
Again, it's the same thing. My guess is probably we can push one more cell cycle, but again, because why I'm telling, one of my data in the mouse heart, where I have knocked out FOXO and FOXO3, I can push the cell cycle one round. Basically, I can show there is increase till 7 days, but after 7 days, again it's coming back to the normal. So even if we have abundant amount of nutrient, I don't think, at least in the present day, I don't think we can make the cardiomyocyte proliferate as compared to embryonic. It's because of the inhibitors. It's because of the unidentified factors. Could be inhibitor way too much expressing, or the activators are not expressing at all.
Okay. From Kanhaiya Lal Chaudhary, what is cardiomyocytes?
Cardio?
What is cardiomyocytes?
Cardiomyocytes are the main muscle cell. Remember our 10 standard muscle physiology, like we have smooth muscle, we have striated muscle, and then we have cardiac muscle. So cardiac muscle, it's a muscle tissue basically, but it's a specialized muscle which is present in the heart and is known as cardiomyocytes. It has a pumping activity. When the cardiomyocytes of the heart pumped, basically the blood comes out of the heart and goes through the aorta and goes to the different parts of the body.
From Abhishek Bardhan, hello ma'am, can you please tell us how did you find out the novel target gene of FOXO and FOXM1? And I'm wondering whether any non-coding RNAs like long non-coding RNAs or miRNAs contribute in this process.
Absolutely. Non-coding RNA, I mean, there will be definitely, if you look in the PubMed, you will see many articles that these non-coding RNAs are regulating, I mean, all the aspect of cardiac biology, starting from development to disease like proliferation, growth, apoptosis, fibrosis, hypertrophy. So absolutely, non-coding RNAs has tremendous role in all the aspects of cardiac biology. And your first question was? Sorry, I forgot.
Yeah. The first question was, can you please tell us how did you find out that novel target gene of FOXO and FOXM1?
See, in order to do that, basically you have to do a lot of literature study and see whether there is any known target in other cell type that is not investigated in the cell type that you are working. That is number one. And if you find that, basically you do some bioinformatics study. There are some programs where they can show you that whether that particular factor can bind to the promoter region of the target that you wanted to have. Like they will give you all the different targets, and from those targets you see whether these target can be one of the potential thing. Okay, then you have to do it in your wet lab. You have to do so from the bioinformatics you can predict, right? But you cannot prove. You can predict, okay, this transcription factor maybe have all these potential targets. Now after going through all the targets, you can select at least 2-3 and do some literature study to see whether those has any role in the known role in the cardiomyocytes or not. Once you have that, then you do some DNA protein interaction by chromatin immunoprecipitation study. You can do some footprinting study in order to see whether exactly this protein is actually bind to the promoter, the DNA region of the target or not. And from there on, you can start. Then you have to do some functional study like luciferase assay to show you the promoter is active or not, those kind of thing. Okay.
So the next is how does, from Subhashish Chandra again, how does oleic acid activate AMPK?
Oh, okay. That we have not investigated actually. We are not sure how, but we think whether it's directly activated AMPK or there is in between, there is something between oleic acid and AMPK, that we are not sure. but that we wanted to investigate actually.
From Abhishek Vardhan again, how does the time span of ER stress contributes to differential results on target molecules?
One is time span and another is the concentration in the sense the extent of insult, extent of damage. It's again coming down to the gene regulation, what genes are getting upregulated, how long the genes are getting upregulated, who is shutting down what kind of genes. Those are the determinant factor.
Okay, the next is from Tanima Sheet. Hello, ma'am. Can I get this PPT?
I can, but there are some unpublished data. So the published data, the arsenic part and the other part you can, but not the ER stress one.
Okay, so from Tanima Sheet, you can directly email Dr. Sengupta.
Yeah.
And Dr.
If you one thing you can do, the student you're talking about, you can type my name in the PubMed. You will get the two papers. One, the proliferation paper that is in circulation research, and then the arsenic paper. It's in cardiovascular toxicology in the last year that got published. So if you read those two papers, you will get the PowerPoint basically.
Yes.
If you are unable to do that, you just contact me. You just email me, no problem. I will share you.
Yeah. Yes. The next is from Anonymous attendee. Heart attacks are more common in men than in women. What's the reason, ma'am?
There can be so many reasons. I think one of the reason is the hormonal regulation. Okay, so basically, and then the stress, the insult, basically the stress or it could be the inherent nature of the cardiac myocytes that is different in terms of men and women. So maybe to answer your question, I think we really don't know the exact reason, but we can speculate. It could be the hormonal regulation, the stress, and the other the inherent nature of the cardiomyocytes.
From Orna Bhattacharya, ma'am, in case of heart attack, is it possible to locally overexpress FOXM1 or inhibiting FOXO in the damaged part of the tissue to proliferate the cell at least for one cycle and so that we can gain back some of the dead cell?
See, these are all the, we need to do a lot of translational research in order to answer all these questions. First of all, we need to make sure that the protein that we are expressing, that will penetrate the cell membrane, and that you're talking about local. So you're talking about the heart itself. There are a few ways by which we can directly inject things into the heart cardiomyocytes, but a protein like a FOXO or some other whatever protein we want, the main problem is the solubilization, and then how we are delivering that, so that needs to be I mean, we need to check all those things.
From Shubhashish Indra, ma'am, can we target FOXM1 and block it by some kind of drug to treat pathological hyperplastic conditions?
FOXM1. See, these are all, I mean, we can activate, we can inhibit. Basically, it's again drug discovery. Okay, so we can't just what we have to do. We have to somehow design a drug or a peptide or a thing that will either activate the target molecule in case if its target is FOXO or your FOXM1. Either you want to activate or inhibit. Basically, the drug discovery is basically we are designing some molecule so that that those molecule can have an effect, whatever effector you are thinking of. Okay, so those kind of thing, basically what we need to do, we need to do a lot of bioinformatics study to see whether these molecules can exactly be either activated or inactivated. There are some, actually there are some FOXO inhibitor and activator FDA have approved, but I don't think anybody has done any human trial with those yet.
Okay. The next is from Kuheli. Can this property of cardiac cells of not proliferating in mature heart be used to treat cancer?
To treat cancer in other cell, you mean other organs? I don't think that would be an ideal thing, isn't it?
Other organ.
It should be, I mean, from your question, I'm assuming that that is your question. Cardiomyocytes will not do anything in other organ, right?
Yeah. So any other question from anyone? So no, I don't see. So yeah, let me check once again. No, I think I covered everything related to this topic. Wonderful presentation and wonderful question answer session. We are so privileged to have you as speaker
Oh, thank you.
in this webinar, and I wish when this corona problem we will overcome these things, and we will invite you to come to visit our institute, and
Absolutely.
we will invite you for another lecture session.
Okay. Thank you so much. Thank you so
very much, Arunima.
Okay,
So I just a second. Another question from Abhishek Bhatt.
Oh, okay. Okay,
Is
sure.
blocking of FOXM1 by MPK tissue specific, or is it ubiquitous?
No, it is, I can't say it's specific in the sense it's not unique to cardiomyocytes. I have tested in cardiomyocytes. That's why I can tell you that it's for sure it's in cardiomyocytes. But since I have not tested in other tissue, I cannot say that it is only in cardiomyocytes. It can be in other tissue types also.
Okay. So any other questions? No, I think if you have further query, you can direct contact Dr. Sengupta, and I'm sure she will help you to understand the subject more. Anyway, thank you very much. So
thank you.
Yes, so I'm ending the meeting for
okay. Should I leave now?
Yeah, please.
Okay. Thank you, Devavani.
Bye.
Talk to you later.